Hello, and thank you for joining us for an Intus Bio Microbiome Geek section with Mark Driscoll, the Chief Scientific Officer, who is going to hopefully enlighten you and blow your minds with all this amazing information he has.
Okay, thank you for that introduction, and thank you guys all for attending this. It's a privilege to be able to talk to you today. What I'm going to present today is the outcome of some of the tools that we have invented. And as Shania said, I am the Chief Scientific Officer of Intus Bio, and we really started from the ground up developing tools to explore the diversity of the gut bacteria and all the bacteria that basically share our living space. So our take-home message at the end of this, hopefully, that you get is that we want to, and we have the ability to, make the microbiome, those bacteria, as integral to healthcare as it is to our health. And the starting point for this is really just the observation when my team developed one of the first, or actually the first, next generation DNA sequencer. I had the privilege of being in charge of that group back in the day. We built the GS20 really to sequence human genomes. But what we found is that we had, for the first time, the ability to see the billions and trillions of cells that are inhabiting our bodies. So it turns out we're actually a composite organism. And there's somewhere, depending on the estimates that people give, between 2 to 10 times more bacteria in us and on us than we have human cells. And there is somewhere on the order of 100-fold more genes in those bacteria working for us. So the idea that you would have a full grasp of human health and not pay attention to this is kind of crazy. But if you can't see it, you can't study it. And we developed the tools to actually take a really close look at this. What we want to do is just introduce the team. So our CEO is here. Here's me. I come from, like I mentioned, this background in next generation DNA sequencing, but other folks on our team as well came along with me in this company to found this. And Dan Fasulo is a key figure. He really is the person who develops the software tools for the analysis and things. And so he was at Celera for the human genome assembly, and he was with me at 454. So we really have kind of an A-team developing this technology. And our fundamental premise here is if you can't see it, you can't study it, right? And that's a basic tenet of science. The other was that bacterial strains appear to drive cancer. We know this for things like Helicobacter pylori, and there was strong hints for things like colorectal cancer and other first world diseases. And with our modern diets and with antibiotic use and first world medicine, we know that those bacteria profiles are changing, and we do inherit those from our parents and from the folks around us. So studying that is important. And then we knew that if we could just get to the resolution we needed, or we figured that would be true, that we could start to develop actionable tests for optimizing your health or diagnosing and preventing disease. And so we do have results around colon cancer that we're publishing and infectious disease, but really what I'm going to focus on today is first world diseases specifically, or for diagnosis and prevention, but for individual microbiome. So this slide just reiterates what I just mentioned, where everything from rheumatoid arthritis to cancer, type 1 diabetes, they've all in some way been linked to the bacteria that we carry around with us on a daily basis, those bacteria that we inherit from our parents and grandparents, and those bacteria that evolve to live with us. Because if you think about it, we have billions of bacteria living all over us, and we're not a raging infection. So there must be some fundamental communication going on there between the bacteria. They're saying, hey, we're the good guys, don't hurt us. And your body and your immune system is saying, okay, I'll let you have some living space. So studying this was really why we developed these tools. Just a little bit of geek background. The reason we're different, and this is published if you're interested in such things in our paper with Jeanette Gehrig and Microbial Genomics back in 2022, we really tried to, there's a number of different ways you can look at the bacteria. Some of the most common are called 16S or metagenomics. And we really developed our platform to overcome some of the limitations of all of these different platforms. And so if you're familiar with this, which most people aren't, 16S is one method. It gives you the ability to see things at low resolution, but a lot of them at once, high throughput. And metagenomics is just the opposite, where you can see things at high resolution, but you can't afford to do many samples. So we really had to cover all of the waterfront here and make sure that this was scalable, and then also high accuracy for actionability. So one of the first questions that we set out to answer was, what does a healthy adult microbiome look like? And what can cause the microbiome to develop problems, and what can positively affect the microbiome? That is, like, what is normal, what is abnormal, and then can you change either of those things? Right, just a fundamental question, because when you're dealing with individuals, you're not dealing with a statistical population, you're dealing with that individual, and they don't want to know, oh, you have a 25% chance of, your microbiome is weird, or they just want to know, what can I do? So really what this presentation is focused on is that. So what does a normal microbiome look like? Well, we have that here. This is, we have our test, which actually uses a scoring system, which I'll explain in a minute. This one was chosen because it's a great score. And so when you look at the first page of your individual microbiome report, it kind of sums up the things that we're looking at. It's an excellent microbiome score. It gives you your enterotype, that basically you can look at that and see what folks have been eating. We look at the different things like the resistome, which is the number and types of bacteria that carry antibiotic resistance genes. We look at pathogens, proteobacteria, which is a pathogen group, fusobacteria, which is a cancer-related bacteria group. So all of these things are called out right up top to see if there's anything peculiar or odd about your microbiome. And just one very important thing, if you don't remember anything else about how the scoring is done, the way we developed these scores was just with reference to our healthy population. So something is strange and different, that is peculiar or out of range, it's all done with the statistics of the population. So we're comparing your microbiome to what we see in all the healthy people. And so this is just a great example of somebody who's so healthy they're breaking a few of the statistics. But what I'll call your attention to is this target plot here. And this target plot essentially is a one snapshot view. You can take it all in with your eyeballs, the entire microbiome in one plot. And so what we do is we actually break this down by taxonomy. What does that mean? Well, we know that all of these are bacteria, so that's in the center. And then we go kingdom or domain, phylum, class, order, family, genus, species, and strain. And if you remember from biology, those are the various taxonomic divisions of the hierarchy of life. So each of these rings tells you something slightly different. But the most important things when you're looking at this, and it seems hopelessly complex when you look at it at first, but I'm going to tell you a few clues, and you'll be reading this as well as I do by about the 5th slide. So let me tell you where I look. The first place I look is at this phylum level, Firmicutes and Bacteroidetes. So they should be, in healthy humans, they're about 50-50. And they should make up the majority of what's in your gut. So this is fecal samples. So when you look at this, there's about 200 different phyla of life and in the bacterial space, but really only 2 of these associate with humans is the majority. Now you'll see over here, there's a few others, and that's fine. I mean, we do have other phyla that are important that associate with us, but they should be at a very, very small level. So that's the first place I look and say, does this person have the normal mix of Firmicutes and Bacteroidetes at about 50-50 and encompassing the majority of bacteria? Okay, yeah, this person does. So then what I do is I skip out to the outer 2 rings. And those outer 2 rings are the species and strain level. And so what we're looking for out here is just a lot of things. So it's a really simple metric, right? How many do you have? Well, the more resilient microbiomes will have a whole lot of things because you've got all these bacteria in here and they're each doing something slightly different and they're complementing each other. They're driving the pH of the gut. So they're doing all sorts of different things, digesting your food and interacting with your immune system. So we find just empirically that, and this has been known in the literature for a long time, that the more of these you have, the better off you are. So I look and see that there's a lot of them. And then the second real question of these 2 outer rings is, are they evenly distributed? More or less, right? So you don't want to see any one thing encompassing a giant chunk of your microbiome, because that means that there's not the diversity that you see in more healthy folks. And so that's really when you read this thing at a snapshot. What's the score look like? Is there anything weird over here that's in any of these metrics that we're looking at? And then does it look like a normal microbiome? Firmicutes to Bacteroidetes, lots of things in these outer two rings and evenly spaced. So relatively straightforward stuff. So then where does the score come from? I need to explain a little bit of that just because when you're looking at these, it's important to know. The score comes from really hard, quantifiable metrics of the microbiome that are universal. So what we found is if you grew up in India, these metrics are going to be the same as if you had grown up in Indianapolis, right? So these are things that are common to all humans, and you might get into down here at the species and strain level, you're going to have a totally different profile of bacteria, but the things we're measuring are really common to all people. So what we do is we look at these things over here under the resilience and biodiversity section in the report when you look in there, but alpha diversity, and these are all explained in the appendix of the report. It's a measure of resilience that is based on the richness and evenness, which, as I just explained to you, is the number of things that are out here in these other two rings and the amount of space that they cover, so that whether they're evenly distributed or not. Beta diversity is another word for how different is this. And so, like I said, this is all statistically derived. And so in this case, there's a flag. It says this one is different because the score is so darn good, right? So this can be good and it can be bad, at least in this case. And in this case, it's a good thing because there's just so much here. The Firmicutes to Bacteroidetes ratio, once again, that's a flag we're looking for. It should be about 50-50. Most people have more Bacteroidetes than Firmicutes. This person has a little bit of the opposite of that. And then we look at things like Fusobacteria. These are the cancer-associated bacteria. They really don't belong in a microbiome. If you see them, that's kind of just a trouble signal. Resistome score, the number and types of bacteria that contain antibiotic resistance genes. So once again, comparing to the general population, do you have more of these bacteria, or are you in the range of 75% of people? And you can see here, it's within the range of 75% of people. Enterotype, that's just, I'll explain a little bit about that later. That's just, you can tell whether you have somebody on your hands who's a vegetarian, who is a vegan, who eats mostly processed foods, or somebody who has a normal Western diet, looking at and also you can tell enterotype for kids. The bacteria that young people have is slightly different than those for adults. Pathogens, this is something, there are known pathogens, right? So we count them up and we say, when you have pathogens, do you have more pathogens than most people have? You know, just a really simple question, because some people carry pathogenic bacteria. They call them pathobionts. It's hard to tell sometimes whether something is a pathogen. It could be at high levels and not at low levels. So we call them pathobionts. But if you have way more of these pathogens, like Klebsiella, E. coli, Enterobacter than most people, then it'll decrease your score. Proteobacteria is another one. It's a phylum that contains E. coli and things. These in normal people are just, healthy people are present at really low levels. You can see here, right? So it's just not much of the microbiome. That's good. And then the last thing that we call out, which is super important, is the top 10 species. So when we look at your bacteria, an important question is, because some of these bacteria do very specific and sometimes very bad things. In your top 10 species, do you have more of something, or is something present at an overwhelming amount compared to what we normally see? So that can be a great clue if he's reporting that they have some kind of symptoms. So that is the explanation of the target plot. And one of the things that we're going to take a look at here is that this particular top 10 species here is an interesting one, and I've highlighted it because it is a small proportion of the microbiome, but it's unusually high. It's at around 4.5%, where in 75% of people, we see it at less than 3%. So it's an interesting one to keep an eye on, because this one is actually named after an antibiotic, and it expands in the presence of certain antibiotics. Leading into that, one of the things we can look at is, how does antibiotic treatment affect the microbiome? So this is one thing that's been known, and what we expect is the assay and the report to behave as one would expect given treatment with antibiotics. So this person underwent, this is the exact same plot that you saw on the previous slide, and they underwent antibiotic treatment. And then we can take a look at how that affected the microbiome. And so once again, keep in mind the top metrics, the score is 100, and the resistome score, the bacteria that carry antibiotic resistance genes, was at a normal level. And then after antibiotics, you can see a whole lot has changed, right? So one of the things that we look at is firmicutes to bacteroidetes. Okay, that looks still about 50-50, that's fine. But then I go to the two outer rings and I see that, oh my gosh, this one bacterium has now taken up about 40% of the microbiome. Well, let's go back over here and take a look. It was just this relatively small slice over here, but suddenly it's expanded to this, and it is in brackets, in the red brackets, it is one of those bacteria that contains or that tends to carry antibiotic resistance genes. This one over here in red is another one that tends to carry antibiotic resistance genes, and you can see also that group has expanded pretty significantly in here. And then the other one, this group that is in green that doesn't really carry antibiotic resistance genes, but they really set the tone for the gut by creating short chain fatty acids oh my goodness, short chain fatty acids and changing the pH of the gut so that it prohibits bad bacteria from gripping hold. These have been completely disappeared. You just don't see them over here. So that's the effect of a single treatment of antibiotics. The score has gone down as you would predict. And then the resistome score, once again, is now pegged at the right, as you would expect, right? Because the bacteria that survived are the ones that tend to carry antibiotic resistance genes. So the microbiome responds as you would expect. The scoring and the behavior of the test or the report behaved as you would expect. And then we can actually build on this a little bit by showing that with a second round of antibiotics with a different one. And I mentioned this over here, small slice of the original microbiome space was taken up by this strange bacteria that is named after an antibiotic. Well, this is the antibiotic that it responds to right here. And so when she took that, you can see that right here, it's expanded as expected to take up a significant chunk of the microbiome. And so you can see, once again, the microbiome score is poor, the resistome score is high, and then these things like P. dorii have grown to encompass a fairly large space in the microbiome. So once again, different antibiotics, different effects, different individual species or strains responding as expected. So we're really able to track these changes in a meaningful way over time. And then what happens over time? So this is where we started. This is where you just saw after the second round of antibiotics. And then this is where we got, where she got after 8 months with no antibiotics. And you can see here, these guys in green, they were a fairly large chunk of the microbiome over here. They're actually starting to come back. So we're seeing a little bit of that improvement, but you can see from the resistome score that it's still relatively high. The number and types of bacteria that have survived really are the ones that carry antibiotic resistance genes, and these really gut-sustaining group of bacteria have not yet had a chance to come back, even after 8 months. So it shows you that with sufficient antibiotic use, you know, it can affect the gut for a long time, even after you're, quote, cured. So what happens even after that? This is after 14 months. Same person. You can see that these guys that were previously suppressed have now come back pretty well. The resistome score is heading back down to a great place. The overall score is good, but still after 14 months, there's still a lot of recovery to be had here. And you can see that this proteobacteria space, which is one you want, it's a phylum you really want to have a small amount of, is still pretty high. So it takes a long time to recover from that, but we can watch it and we can follow it. Switching gears a little bit, this is a different sample. And so what we're looking at here is my microbiome, and it's over 14 months. And the reason I'm showing you this is because when you take a look at a microbiome, the question is it changing so rapidly you can't get any useful information about it? So what I did was I followed myself for 14 months. And what you can see is that it didn't change very much. And then also, if you look at the score, it was terrible. And that didn't change very much either. And if you look at my resistome score, also terrible, and it didn't change very much. And so really this is an interesting, I've just showed you how rapidly the microbiome can change over weeks and months in response to one insult like antibiotics. But what happens if you just look at the same person over time with no changes? There's really nothing going on here. There are no changes worth writing home about. So it's bad, it stays bad. And I just went ahead and lived my life and didn't worry about this. So what I wanted to do then, though, was say, could I change it? And so in consultation with folks, what I wanted to do was just a single thing, a single targeted change. So I knew that it was responsible for the change. I didn't want to do multiple things at once. And what I wanted to do was show that I could move from this to something like this. And how long would it take? And so when I instituted this one change, you could see after 4 weeks, things started to change pretty dramatically, right? So things were shifting around. This giant chunk of the microbiome, that bacterium, suddenly was being suppressed a little bit. You could see my firmicutes going from, once again, firmicutes to Bacteroidetes ratio, right? So you want to see that about 50-50. It isn't. Now it's getting more close to 50-50, which is what you see in healthy people. This giant chunk of proteobacteria is now getting suppressed down to a reasonable level. My score is going up, and my resistome score hasn't really budged much after 4 weeks. There's still a lot of antibiotic-resistant bacteria taking up most of the space. But after 14 weeks, you can see that, once again, looking at the Bacteroidetes to firmicutes ratio, it's pretty good. It's about 50-50. The other phyla have been suppressed down to about where they're supposed to be. That giant chunk of proteobacteria is now a tiny little slice. And I've got a lot of these short chain fatty acid producing bacteria that just really weren't a big chunk of the microbiome. And so the other things, the scores have gone up appropriately, 92 out of 100. And my resistome score has decreased as well. So it really is, there are things you can do that will dramatically change your microbiome for the better. I showed you the worst, I showed you the better. And now that you guys can kind of read this, looking at these charts, I'll show you a couple of interesting changes. So this person here had actually, opposite of me, a firmicutes overabundance. You can see firmicutes to Bacteroidetes. Bacteroidetes is a tiny slice, firmicutes. And overabundance of these is actually associated with weight gain. That's been published multiple times in the literature. And so really what she wanted to do was go ahead and make some changes that might drive these downward and get a more reasonable firmicutes to Bacteroidetes range. Otherwise, perfectly healthy person. And so 1 week after starting the supplement that she was put on, you could see that things were almost the same, right? This doesn't happen immediately, but you can see this little tiny slice here has moved to a little tiny bigger slice here, which now moves to a giant chunk of the microbiome over here. And so these Bacteroidetes, which had been suppressed, are now ballooning outward and becoming a more reasonable part of her microbiome. And so the changes were still in progress as we looked at this, because remember, this is 2 weeks after starting the supplement, and you know, we're going to capture more samples to see where she evened out. But it takes a few weeks for these changes to start to come into effect. So once again, there are some things that happen that aren't good. So this person was had a relatively normal microbiome, but the idea was that, look, the Bacteroidetes were kind of suppressed, and there was overrepresentation of some of these phyla over here. So after embarking on a supplement treatment, you can see that things change, maybe not entirely. I'm going to try something different here, where this one phylum, which is Akkermansia muciniphila, basically this one phylum contains one human-related bacterium. It had ballooned to be more than 25% of the microbiome. And so this kind of thing once again, reading this, right, firmicutes to Bacteroidetes, you want about 50-50. You don't want to see these other phyla at a large percentage. And in fact, you do see this one. So 1 week after starting treatment, we look at this and say, we want to monitor this closely because this is not going in the right direction. It's kind of odd. That's why you need to kind of watch and monitor folks. And then one further example that just now that you can read these target plots over here on the left, you can see, right, so firmicutes to Bacteroidetes, there's only Bacteroidetes, and then look at the outer two rings. Okay, there's only one thing in the outer two rings, right? So it's just, this is looking terrible. This is actually someone who had recently, like the week before, been released from the hospital after being treated for sepsis. And so it was a young person, young kids, actually overall very healthy, ran distance and lifted weights, and just, we don't know how she picked this up. But she did let us follow her samples over the first 3 weeks after coming back, and she told us how she was feeling. And so remember that what the report looks for is, you can see the score is as bad as it can get, of course. And then these little slices in here of things that are present are actually hospital pathogens. But even after September, October, November, December, 3 months, what's come back is, if we're looking at the majority of her microbiome, you can see firmicutes to Bacteroidetes, but not much in either one. And a lot of these are SIBO-related overgrowth organisms. So they're not really what you want to have in your microbiome. They're rare at high quantities, and they are present at high quantities. And then even after 14 months, where she's still complaining of feeling poorly, tired, you can see the score is terrible because what's taken over really are these SIBO-related bacteria. And or these very peculiar bacteria that just kind of, they really don't belong. And also the proteobacteria slice here, which is where a lot of the pathogens or bacteria that don't really interact well with your gut, that's a giant chunk of her microbiome. Firmicutes to Bacteroides ratio is really off. A lot of these firmicutes that you want to be in your gut have been just compressed to almost nothing. So this is somebody who, they said, back here, you're cured. And over here, she says, geez, I still don't feel cured. And you look at her gut and you're like, these bacteria, like it's way off. We need to do something about this. This is just one more here of somebody who just has, once you can read these things, firmicutes to Bacteroidetes. Well, so they are kind of the same. But the problem is these two other phyla are here, and look at this. E. coli in this proteobacteria phylum is a giant chunk of the microbiome. So this will show up in your top 10, and it's going to say hi. It's on the pathogens list, so it's going to trip all these flags that basically say that this is really not a great thing. And you can see here in the metrics, the resistome score is super high, the evenness is poor. So a bunch of these flags will be tripped and let you know that, geez, it's not so great. And then this is just one that was particularly interesting because this is somebody who has colon cancer. And so what we wanted to do is take a look at their gut microbiome. You can see the score is terrible. One of the big attractive things over here is that Fusobacterium nucleatum that you normally do not see in people. That Fusobacteria is just pegged all the way to the right. And once again, this is cancer related, and we do see it in a cancer sample. The second thing that's related to cancer is these E. coli strains, and we can see Klebsiella, basic hospital pathogens, and high E. coli, which are common we find in cancer patients, and that's been found in the literature for a long time. So you just really never want to see the Fusobacterium in somebody's gut. In summary, what I've kind of shown you over about the last half hour is that what we do is we compare to our healthy populations across a bunch of microbiome metrics that are shared across all populations. And a healthy adult microbiome should compare favorably to the healthy population. So what we do is we use statistics to point out the differences in the report. And those differences can actually be actionable. The problems that we reviewed can be high commensals, like in my microbiome, Poor balance, also like in my microbiome, poor diversity. You can have pathogens like I showed you in those last couple of slides, recent antibiotic treatment, which I showed, and also cancer, right? So there are a number of things that are known to affect the microbiome. This test does pick those up. But I've also shown that targeted treatments can be used to push the microbiome around and get, in many cases, closer to the healthy population, but sometimes not, right? You just need to pay attention and see how the actions you're taking are working, actually, with some kind of follow-up. This, I've spoken at length about how we can do this. There are a bunch of partners that we have. I just will point out that we do participate in the InSTAN ring trials every year, and we passed that for the past 2 years. That's basically a European trial where they give us a mystery microbiome. We have to tell them what it is. We have a lab-developed test that we're using with Avero Diagnostics, so that's been through that kind of rigorous but also our publications, of which we have many. A lot of the claims that I make in here that we can detect this or that we're the best at it, those are backed up by these publications. And we have a large number and growing number of collaborators that are using this tool for things like cancer and alcoholism and that kind of thing. So with that, I'm going to wrap it up and ask for questions.
Thanks, Mark. That was great. I especially like seeing the visuals and watching the things change so quickly. It's really pretty amazing. Does anyone have any questions specifically for Mark?
Yeah, Mark, what was the intervention, the single intervention you used to move your ID?
Everybody asks that, and the reason I don't put it in the slide is because I don't want somebody reading the slide and saying, hey, this random guy I saw in a presentation did this, so I'm going to do it too. Like I showed you, like sometimes the intervention the right way, and it just happened to for me. So in consultation with Elena, basically she looked at me and said, what the hell do you eat? And I said, oh, coffee and peanuts. And she was like, yeah, no, not so much. So basically she put me on, she said I needed fiber in my diet. So I chose something off the shelf that's commonly available to everybody. I just grabbed Metamucil and said, I'll use this as a single supplement once a day, 2 tablespoons in water, before I drink my coffee in the morning. And that was the single change that I made. And so the reason I can do that is because, psyllium husk or Metamucil can block the absorption of certain medications. So not everybody can use it, but as a healthy older guy on no medication, I can safely use that thing without any problems. And that's not true for everybody. So that's why that's not in the presentation, but that was it, right? I mean, this basic change you could see, right? So when I went for 14 months without changing anything, I didn't, my microbiome stayed the same. The score was terrible. I didn't really pay any attention to what I was doing. I just lived my life. And, around Christmas time, I was picking out on, all the treats that you get there. And then in the summertime, I was running more than I usually do. None of that stuff had much of an effect. And then, switch to this high fiber thing, and all of a sudden it has a big change. So there are very specific things that you can do in consultation with your nutritionist and the people who know more about these things than I do. So pay attention, because a lot of those things can work quite well.
Thank you.
Go ahead, Poonam.
Thank you. That's so interesting about a simple, small, cheap way to massively change your microbiome. I mean, I definitely think I'll recommend it to patients who are, I guess, well enough to try it. I'm guessing it worked as a very powerful prebiotic for you, so it fed all of your good bacteria, which then could kill off, I guess, all the bad ones, inverted commas. That's so interesting. My question, Mark. So firstly, it was a really good presentation. Thank you. My question is, so I work privately, and I also work for the NHS. I think your very last slide, you said that one of your collaborating partners are the NHS, and I was just interested to know where that might be. Do you know whether it's being used clinically, or is it just in research? And if it is clinically, is it for the colorectal teams? Yeah.
It's contact with the research right now. So we have folks that are evaluating different methods for screening the microbiome, and that's kind of where we're at with that. So we are talking to folks, but as you know, the NHS is broken up over all kinds of different areas, and so we're eager to open up as many of those as possible.
Fine. No, I expected you to say research. I think that maybe in about 50 years the NHS will catch up, and maybe we'll be able to have a sample like this, but at least it's begun, so that's good to know. Thank you very much.
Yep.
Hi Mark, a question from me. Nice to see you again, by the way.
Yeah, nice to see you.
So well, I've seen this before, but a few things I forgot. So as practitioners interpreting this test, you kept pointing towards the first the first sort of level of circle with the bacteroids and the firmicutes. But do we have to know every one of the level, or I forgot what it means, all the different rings?
No, you really don't. I mean, so the basic interpretation of this thing is at the levels that I told you. I mean, it depends on how deep you want to dig, right? So different people will look at different things. So like I showed you, if you really want to get into it, the Lactospiraceae and the Oscillospiraceae, these are two groups that are heavily affected by antibiotics that tend to set the stage in the gut for short chain fatty acid production. But what you can see just simply looking at this is that it's enough to know firmicutes and bacteroidetes should be about 50-50, and they should be very diverse. So when you lose whole regions of those, that's not good. So it's not necessary to know every ring on here. The basic interpretation is really in the upfront of the report itself. So it tells you where things have gone wrong, what to focus on. And then Elena can even speak to the recommended actions that we have in the report that say that, okay, if you see this, then, you can think about these things. And they're really meant for more clinician focus like you guys than they are for individual customers, because, you'll understand more about what those recommendations are. And Elena, I don't know if you want to break in and give a little explanation of that part of the report.
No, exactly. I don't really think that you need to really learn the names of all the species and the strain. This is really not necessary. What you have to be accustomed to is to just look at the picture, just look at the plot, look at our first page, or the summary page, and just look at what is absolutely out of range. So why your client, why your patient is having some symptoms is due to very high pathogen or pathobiome, or even commensal, or even probiotics, okay? So you need to look at any imbalance and then work on the imbalance. And the summary on page 1 is really intended to guide the clinician to make a decision. So it's much easier than that. But obviously, it requires some sort of, experience, like any other kind of test, like any other functional medicine test, for example. The more you see, the more it becomes easier, obviously.
Yeah, and Elena, just as an example, I don't know if you want to address or this is an example of a report we saw yesterday, and I think both Elena and I said the same thing. Look at this, and then what? Allostipes find Goldii as the #1 bacterium. Okay, that's really weird. So I don't know, Elena, if I don't want to catch you off guard, but
no, exactly. Even if you don't know the name of this bacterium, it doesn't really matter. It shouldn't be like that. Okay, it's taking a lot of space and is over dominant. So immediately you can understand. It's very intuitive. If you look at the plot and the color, you can see, okay, there is one single bacterium taking a lot of space. And why is that? Clearly, if it's taking all this space, all the other commensal don't have any other space. Okay, so this is a very intuitive kind of approach. Then, if you want to investigate even further, you can look online if you really want to. And then there are very specific species. And with time, I can actually explain you why some specific species can be associated, for example. For example, I know the allostipes, when they are out of range and they are overabundant, can be associated with neurological problem. and systemic inflammation. So for me, it's pretty easy. But even if you don't know that, you have the medical history. Okay, so don't forget that the microbiome test is not a sort of oracle. It's not going to tell you everything. So you need to combine the microbiome test with the medical history, with the family history even. Okay, so you combine the risk factor, the actual symptoms, and then it makes sense.
And I mean, you've got all the different colors. Does a single color refer to one single species? Like the peach here, it's all got different names.
Yeah. So if you see a color, it will always be that color for that particular species.
What about when it expands throughout the different circles? Like the with the firmicutes, and then the next one is what, Clostridia, and the one after that is
Yeah, so you're right. So the U bacterialis, and then the Oscillosporiaceae. So essentially, these are higher levels of taxonomic classification that you don't really need to pay that much attention to. We colored it like this just so that you could follow this slice down to a reasonable level. Really, once again, what you want to look at is that first slice, firmicutes to Bacteroidetes. Okay, that's a problem. And then look at the other rings, right? So you can see that Bacteroidetes is most of it, and that's mostly 1 or 2 bacteria. So that's a real problem. The other thing is that this person, we saw this for the first time yesterday, which is why I was, it's a little bit unfair to Helena, but we both looked at it. This is a person who came in with unexplained neurological symptoms, and her family has this issue. And so we just looked at her gut microbiome without really any preconceived notions of what we'd find. But it turns out that this particular overgrown bacterium is strongly correlated in the literature over many years with appearance of neurological symptoms. So it's something that could address this. Maybe that could help, but it's a question of cause or effect, right? So at least it gives us something to work with. We can say, okay, there's something going on. We can see something peculiar. That's very different than what most people have. And if we address this, it would change, the gut health problem, but also would address other symptoms over time.
Okay. Another question. I did ask this when the last time we met, but for the rest of the team, really, what test would this replace that's currently out on the market? Would it replace, say, doctor's data stool test, Registra's test, or where does this fit in?
Yeah,
it doesn't really replace any test, if I have to say, in the sense that there is no other microbiome test as complete as this one. So if you're talking about Dr. Data or Genova, the GIFet, these are not microbiome tests, okay? They just look at a very specific number of core bacteria, but not completely the composition of the microbiome, okay? So they measure some inflammatory biomarker, okay, like calprotectin or the short-chain fatty acid, plus a very limited number of bacteria. But the problem is, what if your specific patient has completely different kind of bacteria? You don't see it. So this is why the two kind of tests are really not comparable. So it's not taking the place of anything. It's pretty unique.
So what you're saying is that it complements it, it doesn't replace it. So
yes,
something here, I mean, as a practitioner with, you're faced with patients with a limited budget, and there's possibly something going on with the gut, and I only have money for one test. Where am I going to put the money? I suppose that's my question.
I think you need to follow some sort of algorithm. So what is the main symptoms or problem of your patient? Is the symptom actually pointing out to some sort of parasitic infection? So you're going to go for a parasite kind of test. Or are you thinking about Candida? You're going to suggest a Candida test. But if you have really no idea, and this patient already had all sorts of tests, they had colonoscopy, gastroscopy, ultrasound, and really there is not a real reason for the symptom, and you need to know exactly what's going on in the gut, I would go straight to the microbiome test. No doubt, I would choose this one.
Okay.
Yeah, and I guess the real answer to your question is, what actionable information are you getting out of each test? And so, which one you order is going to depend on whether it's useful to you and the patient.
Yeah, I mean, like we spoke about because one of the questions I asked before was like, well, you're not testing for parasites and other things, and some of the practitioners may find that, like, oh, it's not comprehensive enough. And the question that was put back to me at that point is that how often are you actually getting parasites in the stool? And it's a fair point. Yeah, it's maybe a little bit more in the UK because the average British person actually travels a lot all over the world. They're very well traveled. So we might see it a little bit more here, but then I think it's still probably fair to say that it's bacterial problems that's the core. So it still brings me back to the fact that, I've got a patient complaining of the usual gut type symptoms and the rest of it, and they're like, I want a gut test. And in my head, I'm like, okay, fine. If I were to use this, it means I'll be using this in place of something like the Genoa's or doctor's data, because you have to think of people's budgets. Nothing is everything is self-pay here, so
yeah. And I guess once again it would be where do you think you're going to find the most actionability?
I'm not sure. Like, as I said, I don't have the experience of this, so I couldn't tell, but it sounds like
It really depends on the medical history, because you mentioned, obviously, let's say that your patient went away to a tropical place. Okay, so you have a suspicion that maybe it could be some sort of parasite. You don't spend a lot of money taking a lot of tests, and you go straight to the parasite. But if your patient didn't go to any place, it doesn't have any sign of, I don't know, food poisoning, but it's been having this kind of problem for ages, and there is no answer. What I normally do is usually to combine something very simple like calprotectin. Okay, calprotectin is not that expensive, and you can still combine a microbiome test with calprotectin. Okay, so you know exactly and immediately there is inflammation or there is no inflammation. Yeah, so this is what I normally do. If I suspect something else like Candida, I know exactly which kind of test I have to suggest. But most of the time, this is not the case. And even when there is parasite or Candida, usually there are some sort of bacterial problem and imbalances, because if your microbiome is not balanced, you're going to be much more prone to Candida and parasite. This is my thinking.
Yeah, no, that's true. Okay.
And for this person, basically very healthy, one of the questions she had is, there's something in my microbiome that's weird that's preventing me from losing the weight that I'm trying to lose. And this would tell you that overabundance of firmicutes, which, like I said, is the opposite of what I have in my gut. That's been pointed over the years as, it correlates with gaining weight and keeping it on. So two things, right? Trying to get back to push this back to a more normal firmicutes to bacteria ratio. And then, will that help over time? Well, it takes 2 weeks to start the changes. And then, will you lose weight over time? Well, I guess that depends. But at least, to answer the question, is there something that's visible in the microbiome keeping her from losing the weight that she wants to lose?
All right, I think that might be the end of it. So I'm going to stop recording.